Okay, I would like to thank the organizers for the invitation to speak. I feel privileged to have been given this opportunity to speak in front of you. These are my disclosures. I'm going to give you my perspective as a clinical, I'm not an hematologist, but I'm a clinical pharmacologist, so I'm going to give you my perspective as a clinical pharmacologist, and I've changed a little bit the title, so my title would be more or less, how can we ensure quality drugs for patients with CML on one hand, and how can we ensure that these drugs are still affordable. Now, the global drug expenditure has been rising in an alarming rate, reaching an imaginary figure of $1 trillion in 2013, and according to updated forecasts, it is projected to reach around 1.2 trillion, which is a lot of money, in 2017. At least in part, this remarkable rise reflects our better understanding of the disease etiology, which translates into a rapid or faster development of drugs. For example, a peak record of 36 new molecular entities were launched by the FDA in 2013. Of interest is the fact that the fraction of cancer drugs, here in blue, is continuously growing, again signaling the fact that there is an improved or great progress in the understanding of the pathophysiology of malignant disease. This great progress comes with a significant price tag. At least in the US, oncology was ranked as the top medical discipline on drug spending, accounting for about 9% of total drug expenditure in 2013. Now, one of the most efficient ways to reduce the cost of drugs is the introduction of generic medicines. In North America, in EU, in some other countries around the world, this is possible only after the patent of the original product has expired, some 20 years after it is usually registered. And usually the emergence of authorized copies of generic formulations into the market is associated with a significant decrease in price, ranging anywhere between 70 to 90% of the price of the original product. A recent report has suggested that the use of generic medicine has generated around $1.2 trillion in saving for the healthcare in the United States, which is really amazing to see. Now, the dominant role played by generic medicines, at least in the USA, is also illustrated by the fact that the generic medicine account for about 86% of the volume of prescription, but only of about 29% of the drug expenditure. Now, the dramatic cost of generic medicines is possible because the development process is simplified and shorter because it is based on the experience gained with the original drug, and the regulatory requirements are really much simplified. Now, the regulatory framework for the approval of generic medicines, chemical generic medicines, has been laid down on 1984 in the modern era, by what is called the Drug Price Competition and the Patent Restoration Act, also known as the Hatch-Waxman Act in the United States. Now, in order for a copy of a chemical compound to be approved for marketing, it should fulfill two requirements. First, it should consist identical, not similar, but identical active ingredient, dosage form, and strength, and to be administered via the same route of administration. So this is the first thing. Now, a drug that really fulfills these conditions is considered to be pharmaceutically equivalent, but this is not sufficient. The copy drug also has to exhibit a similar pharmacokinetic behavior, which means basically similar extent of absorption and rate of elimination. This is done by comparing the maximal plasma concentration and the area under the curve, and this is a little bit more complicated, so I'm going to go through that a little bit in more details. Let's imagine, for example, that this is the plasma concentration after a single dose of a drug. The maximal plasma concentration, here in yellow, is one of the parameters that we are comparing the reference with the generic formulation, and we want it to be similar. It is a marker, mostly a marker of absorption. The other parameter is here the shaded area under this curve, which is the area under the plasma concentration time curve, also known as AUC, is a marker of absorption and elimination. Now, we understand, we acknowledge the fact that some minor pharmacokinetic differences may exist even between different batches of the same drug. So as long as these differences are within certain margin, this is still okay when you compare pharmacokinetics of generic to the reference drug. Now, a drug that, a copy drug that has a similar pharmacokinetic behavior, that means similar AUC and AUC and Cmax, is considered to be bioequivalent. And according to this paradigm, which we are using all the time, a drug that is pharmaceutically equivalent and also bioequivalent is expected to be therapeutically equivalent. That means to exert similar or almost identical therapeutic effect and to have similar rate of adverse effect. Now, manufacturers of generic medicines are not required or exempted actually from performing long and expensive phase 3 studies which largely explain the decrease in cost of these generic drugs. So many thousands of drugs have been approved throughout the years and were introduced into the market. Some 30 years later, we can really conclude safely that a drug, a generic drug, that has been approved according to these guidelines, but according to these guidelines, is as efficient and as safe as the original innovator product. This is, for example, a study that compared the clinical efficacy and the safety of cardiovascular generic medicines. The study included some 30 different randomized control studies involving more than 800 patients covering 8 different subclasses of cardiovascular drugs. And as you can see in this slide, the generic medicines were clinically equivalent to the branded drugs. And this is indicated by the fact that the point estimate for the comparison, these black dots, lies near the 0 line, meaning that there is no advantage in favor of either the brand or the generic formulation. Now, despite this positive cumulative experience with generic medicine, the introduction of a new generic medicine is often unwelcomed by the patient and the medical staff alike. There are several reasons for that. One reason is the fact that there are multiple editorials or commentaries that are published in international recognized journals. And for example, let me tell you of one study that compared, evaluated 43 such editorials on generics. And in 50% of these editorials, negative views were expressed against generic substitution. So this is one thing. There is a general atmosphere against it. In addition, the patient perspective is quite obvious. The patient perceived the switch to a generic medication as an inconsiderate administrative decision that is motivated by financial reasons on the account of the patient's well-being. This is quite common. Now, the third thing, which is one of the most important contributing factors to this negative approach towards generics, is the fact that there's lack of awareness among patients and medical staff about the strict regulatory guidelines used, at least in North America and EU, regarding upon which these generic medicines are being really approved. Now, in addition, in many cases, the patients first discover that he's been prescribed a generic medicine only at the pharmacy level, without getting any proper explanation from his treating physician. So the medical staff, including myself, we have a critical role in providing our patients with reassuring explanation about the clinical equivalence of the generic drugs that we prescribe to our patients. If this process is ignored, and I'll tell you an example in a minute, the patient might feel that he's anxious, he might feel that he's been prescribed a low-quality drug and might stop the drug altogether. And my own example is taking care of my own mother, and all of a sudden, one of these days, I'm bringing her medication, as I do always, and she comes back to me within 1 minute, and she tells me, this is not mine. And I would ask her, why is that so? Because it's different color and different name and different, etc. And I forgot to explain her that this is just a generic switch. So I come and I explain it to her, and she lives with me. And after 2 minutes, she looked at me and says, okay, it is okay to say that you have made a mistake. I said, no, it's not a mistake. It's the same drug. You can read. It didn't help. And this is for a patient who lives with a clinical pharmacologist at home, you see? So what happens if you don't have a clinical pharmacologist around you? Well, usually you would throw away the drug. You won't use it. So we are not doing a good job in explaining this to our patients. Now, finally, in many countries around the world, the approval of generic medications does not follow the accepted international guidelines. This is really quite common in many other countries around the world. And this is usually creating low-quality drugs which are most likely therapeutically inferior. And the availability of such low-quality copy drugs has created an indiscriminate public negative publicity against the generic concept as a whole. Now, how all this is really related to CML patients, and I'm going to give you an example of Imatinib. Now, the approval of Imatinib Revolution, the prototype of the tyrosine kinase inhibitor, also known as Gleevec, on 2001 by the FDA and later on by the EMEA, has revolutionized the treatment of CML patients. You know that from your own experience. Now, the updated results from the IRIS study indicate that 85% of the patients are alive after 8 years, and this figure even rises up to 93% if death causes that are unrelated to CML are not considered. So this striking improvement, this is really a striking improvement over the 5 to 6 years of median survival in the pre-Imatinib era. In addition, in 81% of the patients, there were no signs during these 8 years of CML recurrences. So these stunning results really represent a dramatic improvement and dramatic change in the course and the prognosis of the disease. And Imatinib has created a new hope for patients for a better life and longer life with a relatively few adverse effects. Now, despite this enormous success, the penetration of Imatinib drug is rather disappointing worldwide, being about 30%, mostly because it is high cost, mostly. Now, the patent of Imatinib has expired on 2013 in Europe. Soon after, 4 different generic formulations of Imatinib were approved by the European Medical Agency. In addition, 2 additional generic formulations were approved by the Canadian authorities in 2013. The approval of these generic formulations was done based on the demonstration of pharmaceutical equivalent. That means the active ingredient was found to be pharmaceutically equivalent and demonstrating in a bioequivalent study of similar Cmax and similar AUC. And this is really illustrated, for example, here for one of the formulations that was approved in Canada. And as you can see, in yellow, the generic formulation, in red, the Gleevec, and you can see there is almost identical Cmax, almost identical AUC. So based on the paradigm of generics, on the approval of generics, I can say quite safely that this drugs would be therapeutically equivalent to Gleevec. That means that they will exert similar therapeutic effect and will have similar rate of adverse effect. Let me touch on one important issue regarding imatinib. Despite the great success of imatinib, about 10 to 15% of the patients fail to respond or relapse after initial favorable response. There are multiple reasons for that. Among these reasons, the emergence of mutations, the problems with compliance, of course, the stage of the disease, its presentation, and its severity, and some issues regarding pharmacokinetic variability. And in this regard, there is a unanimous agreement that the higher the trough plasma concentration of imatinib, let me explain what is trough plasma concentration. This is the plasma concentration just prior to the administration of the next dose during chronic administration. So the higher the plasma concentration of imatinib, the higher chances of response. This has been really agreed upon all the researchers in this area. In addition, there is also several studies suggesting that a trough plasma concentration during multiple doses, during chronic dosing, of above 1,000 is predictive of better clinical response, attaining of molecular response, and a cytogenetic response. For example, a sub-analysis of the IRIS study has found a significant correlation between the plasma trough concentration of imatinib during chronic dose administration and the chances of acquiring, of attaining complete cytogenetic response. So the higher the plasma trough concentration, the higher chances of getting a complete cytogenetic response. And this is shown here. For example, for those with the average mean, which was low, only 76%. For those who had intermediate levels, 85% obtained this cytogenetic response. And for those with a higher plasma concentration, 92%. So this is clear. Now the question is, in addition also there is this data suggestion from the same study that the mean plasma concentration of patients who achieve the complete cytogenetic response was around 1,000 in red and significantly higher than those who did not achieve this response. So the question is, should we be concerned about that? Meaning, as I explained before, the approval of generic formulations in the EU and North America and Canada was based on a crossover study done in 44 healthy subjects demonstrating bioequivalence, meaning similar Cmax and AUC. But since this was a single dose study, of course, trough plasma concentrations were not compared. And remember, trough plasma concentrations are the best predictive of response. This was not compared in the approval process of generic formulations. Should we be concerned about that? And here is what I would say, that the plasma trough would be after 24 hours, that is after just prior to the administration of the next dose during multiple dose administration. Well, from the scientific point of view, and this is really important, well, the answer is that we do not have to be bothered. That is, if two generic formulations or two formulations of imatinib are judged to be bioequivalent according to the accepted guidelines based on a single dose study, they will have similar trough plasma concentration during multiple dose administration. So this is something that although it's not examined, it should be the same. Nevertheless, both the EMA and the Canadian authorities have demanded that a risk management plan should be in place at the time of approval. And even in the Canadian authorities have required an intense monitoring of the oncogen transcript done every 3 months during the first 12 months after the switch, even if the patient has been stable for many years. So this is something to tell you that we think from the theoretical point of view that should be the same. Still, we would like to be cautious. Now, since clinical studies are not part of the mandatory requirement for the approval of generic medicines, we do not have any clinical data on clinical efficacy or safety of the approved generics in the EU and Canada. We do have some case reports describing relapse after switching from Gleevec to copies of Imatinib. Now, the value of this report is rather limited. I tell you why. It's limited because of their anecdotal nature, and also because in these papers, on this report, it doesn't provide data how this copy of Imatinib, on what ground they really got approval for marketing. So we don't know what was the quality of these copies that were used. Let me just remind you that in some countries around the world, you can get approved generics only based on in vitro studies. That is, in the lab, without any clinical studies, based on what we call in vitro dissolution testing, without a bioequivalence study. So we don't know. On the other hand, we do have some small comparative observational studies regarding generic formulations of Imatinib. For example, one such study that was recently published, actually it is in a preprint status, described the use of 3 generic formulations of Imatinib that have been in use in Turkey since 2012 among 145 CML patients. Now, despite the short-term follow-up of about 12 months, the authors concluded that the molecular response is quite similar between those who remain on Gleevec and those who were switched to the generic formulations. In addition, the mean weekly dose of Imatinib in both arms was similar, which means that the tolerability of the Gleevec and the generic formulations in this case was probably more or less the same. This is only 12 months. There is another study that comes from India that actually compared the response to Gleevec to an unknown formulation of Imatinib among 213 CML patients. Now, the patient in the Gleevec arm got it through the GPAP initiative, so by definition, they were of low socioeconomic status. A larger fraction of patients in the generic arm got actually complete metabolic response, which reflects probably the fact that the patients in the Gleevec arm presented in a late stage, in a more advanced stage. In addition, the clinical response rate was quite similar, with some small non-significant advantage in favor of the generic form. Now, the cost of Gleevec around the world varies greatly, ranging anywhere between $2,000 for 1 month of therapy in Russia and up to close to $8,000 in the USA. Now, the cost of the copy drugs, just for your knowledge, in India is between $150 to $200 for 1 month of therapy. The cost of the generic formulations that were approved by Canada and the EU is currently unclear, but it's supposed to be around $500. So, which means that there is about 80 to 90% cost reduction. Now, the high cost of drugs, of cancer drugs, and in particular of tyrosine kinase inhibitor used for the treatment of CML patients, has been listed as one of the major reasons for the reduced penetration and for the low adherence of these drugs. Now, the newly approval of this generic formulation is expected to markedly enhance the penetration of Imatinib and hopefully also to enhance the adherence. Now, coming back at the final slide of my talk regarding how can we assure the quality of the drug, let me first of all state that the approval of generic Imatinib, if done properly, should be welcome. Now, approval of generic of Imatinib should be permitted only if it is accepted according to the accepted guidelines, meaning for drugs that are pharmaceutically equivalent and bioequivalent. Now, since the trough plasma concentration are not part of the comparability exercise that's done during the approval process, approval of generic imatinib should be combined with the requirement, mandatory requirement for clinical and laboratory monitoring, intense laboratory monitoring during the initial period after the switch. Now, patients or physicians that are taking care of CML patients should get familiarized with the concept of generic and how they are approved, so they can provide their patient with the appropriate reassuring explanation regarding these newcomers. Now, concerning patient advocacy organizations like yourself, I think they should take active part in this empowerment process, thus providing CML patient with the appropriate information and data to ensure that adherence is not compromised due to misconception. Thank you.
Thank you, Professor. We are open for questions. We have 5 more minutes. Edward.
My name is Edward. I've followed this discussion very well, but probably I need more explanation in as far as generics are concerned. Is it a blessing or a curse to the patients? Because initially when I heard about generics, or according to the information I have, the basic information I have, it's like administering, should I say, poison. It's like, when I first heard about the generics, it's like, it shouldn't be given to any person. In my question, I ask, is it a blessing to the patient's community or a curse? How can should a patient accept to take those generics? And now that I don't understand, I need a brief explanation about what generics are, and if they don't follow the international recommendations, then why are they accepted to be given to patients? I'm kind of need some explanation.
Right. Should we have some more questions and answer them together maybe, or?
Do we have any more questions? Oh, many.
If you can touch in a few words, I think that many of us, including me, do not understand the difference between crystallization of different drugs. For example, we know that the Teva generic is crystallized by alpha or beta, I don't remember, and the Novartis original is crystallized by the other one. So should the patients be aware about differences between this crystallization?
Right, so let me answer first of your question. My view is that as long as generics are approved according to guidelines, to accepted international guidelines, they are a blessing. Why? Because they make the drug more affordable. And then, it's possible to give it to many, many more patients. Otherwise, it's really very expensive. But it has to do with the way the drug has been approved. And patients do not have, I think, the knowledge to understand really whether the drug that was given to them is really a good or bad drug. So this should be left, I think, to experts and to the organizations, so that, for example, it is in my view, it is the duty of the country, of each country, to make sure that generic drugs are approved and get marketing authorization only if they are done properly. If these generics, and there is a misunderstanding, if these copy drugs are not approved appropriately, well, they might contain less active drug, or not with more impurities, with differences in the way they are behaving, and the therapeutic effect would be different. So the answer in general, yes, but we have to make sure that these generic drugs are approved according to the guidelines. So this is very important. in any one of these each nations around the world. So it's a bit different in different parts of the world, of course. So now coming back to your question, Giora, this is really complicated, but the way a drug crystallizes differs. It can really be, and now, for example, in imatinib, the Gleevec crystallization form is called beta crystallization. I've got some very nice slides I can show you. For someone who is not an expert in the field, it doesn't mean a thing. The question is now, the generics are alpha crystallized. Now the question is, does it translate, that this different crystallization translate into different pharmacokinetic behavior? Which this is what is really important. And the FDA has a specific guidelines explaining when does it matter and when it doesn't matter. So when there is different crystallization, you have to take care that the dissolution, that means how the drug is dissolved, does it differ in this different crystallization? Is there differences in this?
And the generics are not equivalent to the originals.
Is it working? Yes. So, you have to be very careful with these statements. As a whole, generics, and if you look into the literature, it's hardly, you can hardly find any evidence that generics did not function well. There are some instances, but whenever it happened, it was done because of non-compliance with these regulatory requirements. So when you do some discount for this requirement, you end up in disaster. So this is like, for example, in the L-tyroxine story and some other stories. But in general, thousands of drugs, generic drugs, have been approved and they're all working well. Now the patient, and this is the example of my mother, I didn't tell you the entire story. After 2 days, she came to me and she said, you're telling me it's the same drug, but I feel horrible. She really feels it's the same drug, the same active ingredient. 2 weeks later, everything is gone. So there is some, I guess, psychological effect to the fact that you're used to take this drug, the way it looks, etc., and all of a sudden you have another drug, and as long as you give explanation and as long as you find out that this was approved appropriately, I think this is okay, and the same is also, I think, for the imatinib. This is my view at least.
Okay, we have one question in the
Professor,
back and then
I got
Bert.
2 questions. One is, you mentioned about equivalent active ingredient. For instance, Gleevec, if somebody after the patent expires, some companies come up and use the exact ingredient, and what do you call it? Is Gleevec equivalent in this case? Do you still have to go through the similar trial?
Right. Yeah.
Like
yeah. You have to prove
one.
first. Sorry. Go ahead.
And the other one is like what Gloria was saying. You have different form of similar substance, but because of this molecular end groups are different, but it has similar effect. Then in this case, I guess you have to go through a thorough test. So in this case, how long normally do you need to do this test? Because it's always difficult if it's done centrally for government, in this case, to recruit members, if you're trying something new.
Yeah.
Compared to, it's just an extension of, because the patent has expired, so some companies do exactly use the same active ingredient, so you can skip that step. So,
right.
yeah.
So let me just briefly answer you because of the time limitation. Nowadays, with the sophistication of the laboratory, etc., it's doable to show that the active ingredient in the generic drug and the active ingredient in the original drug are not or are identical. They have to be identical. And there are what we call fingerprint of the drugs. So it couldn't be, just a little bit similar. It should be identical. And if it's not identical, it's not going to be approved by authorities around the world. So this is the first thing. You have to do it. Now, when it comes to biological drug, it's a bit different. And this is not a biological drug, but then you have a lot of problems because there is no way you can produce a biological drug that is really an exact copy of the original drug. But this is a different story. This is a chemical compound. Now, the fact that it's crystallized differently, for example, in this case, it means that you have to do some extra test in the lab to see whether it affects the way it dissolves, the way its stability, etc. And there are some specific guidelines how to do that. So I guess this is, in short, the question. Of course, it's much more complicated than that. For example, there is different analytical methods, and you have to use the right analytical methods and not a compromised one.
Bert?
Yes, my understanding is that sometimes actually the company who makes the original drug and loses a patent also might own other companies that then sell basically the generic medicine under a different name. Is that basically is there sometimes the case that that medication then basically just relabeled and then sold under a different brand, and it's basically coming from the same factory line?
Yes, this is, I guess, an unfortunate, I think, process, because according to the regulations, the first company that produced the first generic can get exclusivity for 6 months, at least in the North America and the EU. And once this is done by the original company, so this is prevented, you see? So they cannot be the first one. And this is, I think, a mistake, because you want all these generic companies to spend time and to spend effort to produce this generic medicine on time, so we can introduce them into the market appropriately once it is possible. But the process and the phenomena that you've just described is really there. I mean, there is nothing much you can do about that. It's really happening all the time. Very hard to follow that. Okay.
Yeah, I would like to continue, but we still don't have the monitors working, so maybe you should answer. There is one question there also.
Sure.
Thank
I'm
you
really
very
sorry.
much. Thank you very much, Professor Caraco, for introducing us in the problems of generic drugs. It is very important for most countries in Europe as well. I came from Poland, where the problem of generic drugs is very actual. But my question for you is following. I would like to know your opinion about the impurities occurring in the generic drugs and also in original drugs. In my opinion, and I hope in your as well, the contamination of two drugs are very important for the quality of the products, for the toxic of the products. Please short comments in this matter.
Sure. This is really a very important issue. When I say that you have to show pharmaceutical equivalent, there is a list of at least 25 different tests that you have to show to the regulatory agencies. It's not only that the same active ingredient is the same, identical, but also that the amount of the impurities, what are their nature, what is the concentration, is there any metabolites that are different, etc. And each one of those is a section in the requirement. And this is really done very meticulously, at least in the EU and in North America. Unfortunately, I'm not sure this is done properly in other parts of the world, but at least in North America, in Europe, it really, now, when it comes also, and I'm going back to the issue of biologics, the impurities in biologic is very important because they may create antibodies or immunogenicity, and then you have to be very careful because then you may create a real problem. It's not only a question of efficacy, but also a question of safety there. Okay.